Iorana, bonjour. Thank you. So I'm going to present to you today some work that we've been doing on Rettis excellens. I feel quite a lot of pressure now after the welcoming speech to show you how exciting excellens are. Hopefully I can convince you a little bit. And this is work I've been working on at the University of Otago with Anna GOSLING and with MATISOO-SMITH, who will be presenting some related work in the series of this conference. You have heard a lot of talks already this morning that have pretty much outlined the rationale behind this research. We are, of course, aware of the timing around the settlement of the Pacific, and we can construct some figures such like these which show general direction, but we all know that it's a lot more complex than what an image like this shows. Rather than nice kind of movements moving from one island to other without any post-settlement interactions. So that was one of the rationales behind this research, was to try and better understand some of those post-settlement interactions, try and look at the origins of the Lapita peoples, and also move more into Polynesia, close to our home Aotearoa, looking at the settlement of Aotearoa and the Chatham Islands, Lekohu, as well as elsewhere in Polynesia. So that was the main driving force behind this research. Many of you will be probably well familiar with the commensal model. We're hearing Lisa having talked about it at previous similar conferences to these, but this is based on the idea that, of course, the people carried with them important plants and animals and introduced them to the lands as they arrived. So animals, we're looking at the Pacific rat, Rettis excellens, chickens, dogs, pigs, as well as a number of plant species as well. So these were, of course, animals that couldn't have arrived in a new island without the transportation by the humans. So therefore, this is based on the idea that if we can look at the genetic data from the animals that humans were carrying, we can begin to understand more about the humans that were carrying those animals and possibly the origins of those humans by proxy. So this particular piece of research focuses on the Pacific rat, Rettis excellens in particular. So excellens, I'm sure again you're probably well aware that excellens was carried intentionally. So unlike the European rats that have arrived on these islands, like the Norway rat and the ship rat, which were stowaways, the Pacific rat was carried intentionally. And could not have moved across the water without being on the waka of people as they moved. And much of the earlier work, like all ancient DNA studies, working with the commensal model, looked at a short region of the control region, so only about a couple of hundred bases. These days, of course, we can sequence the whole mitochondrial genome if we want to. We can even sequence nuclear genomes from ancient DNA. So this gives us access to a much higher resolution data set than what was possible with only a short region of the control region. Nuclear markers, of course, give us the ability to look at both maternal and paternal ancestry, so that can kind of inform us a wee bit more about the movement of these animals. So Lisa's earlier work on the using the control region of the Rettis excellens identified three main lineages. One was widespread across much of the Pacific. You can see there's wee triangle image logos here. One kind of really only as far, not much further than the Solomon Islands, and another more restricted lineage here. And so that was using about 200, 250 base pairs. We had a master's student in our lab a few years ago now, and she looked at the complete mitochondrial genome of some, she was mainly looking around Rapa Nui, Easter Islands, kind of some East Polynesian sites mainly. And this really highlighted to us the fact that we can get a lot more information out of the complete control region. You can see we're already being able to split apart that one lineage that was found across most of the Pacific and look at multiple different haplotypes using the complete mitochondrial genome. So that provided inspiration for this study, and also the idea that we could use this to look at some of those kind of more immediate origins for the settlement of some of these islands. So we have got kiore at Rettis excellens from across sites from right across the Pacific, and we're using a mixture of modern tissue and ancient DNA. And so the ancient ones are shown here in black. You can see quite a few across here. We're always happy to talk to people if you know of any other ancient samples that are around. So catch me if I'm not chasing the baby during the lunchtime. And we've also got modern samples from across the Pacific as well. So with the Rattus excellens, they're probably a wee bit different to some of the other commensal species in that they don't interbreed with some of the introduced, the animals that have been introduced since European settlement. Of course, modern chickens, it's a wee bit hard to tell whether this was a chicken that is a direct descendant from 600, 700, 800 years ago, or has been breeding with European chickens. So the rats are a wee bit unique, which is kind of cool. So this is the classic situation you get in a conference where I think it's quite difficult for you to see. So I'll try and talk you through this one. So we've got a haplotype network here. For those of you who are unfamiliar, the size of the circle is proportional to the number of individuals that share that haplotype. So a haplotype is just a specific set of DNA sequence that is inherited together, if you like. The little black circles represent missing haplotypes, and the dashes represent one-step difference, if you like. And the colours represent different populations. So I'll try and talk you through this because it's quite, it's beginning to get quite exciting. This is still a work in process, so we still have to complete this. But what we're beginning to see is pretty interesting, I think. So if we have a look at perhaps some of the East Polynesian sites, what we can see is we've got two distinct clusters here. So we've got this cluster here, and I've put the locations here in text, so hopefully you can read that if I'm talking too quickly. The ancient samples are underlined. So we've got a bunch of samples from here: Aotearoa, Society Islands, Cook Islands, Mangareva, and Marquesas, all clustering together here. Which is perhaps what we would expect to see based on the geography, based on what we know about archaeology and some of the human genetic data as well. But interestingly, we've also got this other cluster here that is quite distinct with ancient and modern samples from Lekohu, from the Chatham Islands, just near New Zealand, and from the Marquesas and Rapa Nui here. So I think that is pretty cool. The Chatham Island question was one that we were hoping to address because based on some of Lisa's earlier work, she had identified that there was some defining SNPs, some defining mutations, if you like, that set the Chatham Islands kiore apart from other kiore in her research at that time. So we were hoping that the complete mitochondrial genomes might be able to help us tease that apart and look at the origins of the Chatham Islands kiore. I'm sorry, I keep on saying kiore, and I told myself I was going to say Pacific rat or excellence. So just going back to our network here, we can see that indeed this has held up with the complete mitochondrial genome, and we do have this distinction of the Chatham Islands kiore, for example, from mainland Aotearoa and from elsewhere and in East Polynesia. But interestingly, very close to these samples from Marquesas and from Anakina, from Rapa Nui in here. So that's pretty cool. This is quite new data, so this is the part that drew my attention first, possibly because it was close to home. We've then got a wee cluster here from elsewhere in Polynesia. We've got New Caledonia, Samoa, Fiji. This is at a Kimba site in Tokelau. And then interestingly, over here, we've got some Rotuma samples, so also Fiji, but we know from some of the archaeology and from the humans that they can be different to elsewhere in Fiji. So that's pretty cool. And clustered also with a sample from Puka Puka. So Cook Islands again. So the modern Cook Island samples are over here. This ancient Puka Puka sample, again, we understand is likely to be different to elsewhere in Cook Islands. And so we've got this wee cluster over here, which is kind of cool. Another aspect that we were hoping to address, of course, as I said, we were using ancient and modern samples, and that's based on the assumption that there hasn't been turnover through time, and that the modern samples will still be representative of the samples that arrived when people first arrived. And we can see that's probably true for sites like the Chatham Islands. We've got the modern samples don't have a black circle around them, and the ancient samples do have a black circle around them. And we can see they're very similar which is probably what we'd expect with a site that was settled so recently as the Chatham Islands, and Samoa, which at this stage is the only site other than the Cook Islands, which is a wee bit different because this individual is from Puka Puka. In Samoa, however, though, the ancient Samoan is looking quite distinct from the modern Samoan sequences that we've got so far. So it'll be really interesting to fill this out with some more modern mitochondrial genomes and see how that compares across some of the different sites. So, like I said, we're still working on addressing this aspect, that there is little to no genetic turnover through time. But based on this assumption that there is little to no genetic turnover through time, because we can look at the modern excellence, we can then have access, I guess, if you like, to a greater genetic data set in terms of we can better access the nuclear genome. Nuclear DNA is pretty hit and miss, as you're all aware from ancient DNA, and particularly ancient DNA in the Pacific. So we're working on generating some low coverage whole genomes from modern excellence. The first step in doing that was to work with some collaborators in, of course, sequencing the genome so that we had some way that we could assemble these genomes. So we're at that stage now. And then looking at some nuclear regions of the from the ancient samples where possible. And we do have pretty good preservation from the excellence across the Pacific. I was quite surprised. When we compare it to the chicken data, for example, the chicken data has not been as good as what it could be. But we've had pretty good preservation, I think, from the excellence across the Pacific, which is pretty cool. So we're still unable to fully address these questions. And I think as well, excellence alone aren't going to be the only thing that's going to help us to address these questions. We need to be taking a holistic approach and working with everyone here, I think, looking at how the archaeology, the linguistics, oral traditions, and genetics can all kind of intertwine to help us better understand what was going on in this region as people were settling the islands. I just want to acknowledge that this work has been supported by a number of iwi, or a number of New Zealand tribes, New Zealand Māori tribes, sorry. I've had help with the lab work and also with the looking at the complete genome. I have, of course, forgotten to add collaborators who have helped us access different excellent samples, and we're really appreciative of the people who we have worked with so far. And hopefully I'll get a chance to chat to some of you during the breaks.